So I don't want to just talk about early life to you today. I want to put it into the perspective of put it into a spatial perspective and also in a temporal perspective, and show why the study of early life is really important in the search for extraterrestrial life. And the reason for this is, if I can get, yes, there are so many ongoing missions, future missions, looking for life in one way or another. This is a very busy slide. It shows in situ missions on Mars, future missions, possible missions to the icy satellites, as well as the telescopes that are looking for habitable planets and possibly biosignatures. What are they looking for? Well, most of them are looking for some kind of molecular signature of life. On Mars, for instance, the molecular traces of life, possibly even fossil traces of life, such as these Early-Archean, Paleoarchean stromatolites. And with the future sample return mission, possibly even traces of fossilized cells. But mostly, whether it's Mars or the other planets in the solar system, we're looking for the molecular traces of life, or trying to understand the molecular carbon. But what exactly does it all mean? So this is where I switch back to Earth and to put early life into the perspective of the evolution of life. So with this slide, on the left, you have the timescale from the consolidation of the Earth about 4.56 billion years ago to today, with the various geological epochs. And the key factors in the evolution of life over this period of time: the emergence of life, we don't know when, but sometime in the Hadean, the development of early life, of great importance, the emergence of organisms able to split water to reject photosynthesis, oxygenic photosynthesizers, that led to rise of oxygen in the environment, slowly in the atmosphere, in the upper layers of the water. And all of this led eventually to the emergence of eukaryotes, increasing oxygenation of the oceans, until this period where at the end of the Precambrian, the Neoproterozoic, a great spurt in the evolution of life that you can see here. We can look at it in this respect. Today we have life that can be found in every single habitable environment, from the deep crust, microbes and microorganisms in the deep crust, through to the land, to the air, to the sea. Everything is inhabited, and we also have technologically capable bipeds. Now, I want to put the evolution of life on Earth in another perspective. This perspective is using a very nice sketch from the NASA Astrobiology Institute that I like very much, showing the evolution of the consolidation of the Earth, the prebiotic chemistry, evolution of life, and coming up to the life on land, up to us with telescopic observations, etc. And you'll see in the why I'm using this and putting it on a horizontal scale. So we have the emergence of life somewhere between whenever there was liquid water at the right temperature, between 4.4 and 4.2 billion years old. We have the oldest traces of life, the oldest traces of life known are not the first traces. We don't have record of that. These are the oldest preserved traces, going into oxygenic photosynthesizers with the great oxidation event about 2.4 billion years ago, when oxygen started rising in the atmosphere. And so through to the evolution of eukaryotes, diversification of life, and up to mammals. Now, the important thing about this, all of this evolution, is that, and this is what I'll be talking about, but the important thing is that it's all related to the geological evolution of the Earth. For instance, life emerged on a planet, a one-plate planet probably, or maybe with some kind of plume tectonics. It's not like the planet Earth today, where we have very active plate tectonic situation. The oldest well-preserved crustal remnants occur in this period, where we have plume tectonics, or some geophysicists talk about slushy plate tectonics, and I think Lena Noack will talk about that a little bit later. I've seen her name on the list. Which eventually developed into shallow plate tectonics, where we start getting crustal growth, growth of continents, and also nutrient cycling. And this happened about as of about around 3.2 billion years ago. And this is when shortly afterwards, when oxygenic photosynthesis may have appeared, and all associated with the growth of continents, plate tectonics, the area, the habitats available for photosynthetic organisms. Oxygen was able to creep up and oxidize also the atmosphere. And then for a long time, nothing happened. The in-between period, most of the Proterozoic is called the boring billion, because nothing much happens. But then about 800 million years ago, maybe earlier, there's a lot of debate about this, but plate tectonics, as we know today, seem to have been initiated. At the same time, the Earth, there were a number of periods when the Earth iced over. We have the snowball Earth, and this is when life diversified completely. So what we have is co-evolution of the Earth and life, and this is really typical of the Earth, and it has a strong effect on what the kinds of life forms that we have on Earth today. So let's go back to the beginning and look at global conditions when life emerged on Earth. Life emerged on a rocky planet. The rocky planet is very important. It emerged on a planet where there were water bodies with a suitable temperature for the molecules to do their business, get together, where the prebiotic reactions could take place at about temperatures, let's say about less than 120 degrees centigrade. It emerged on a planet where there were oceans and protocontinents, not continents as we have them today, but continents. Well, the protocontinents were something similar to present-day oceanic plateau, like the Ontong Java Plateau or the Kerguelen Islands, or even Iceland. They were small, very small, mostly submerged underwater. Life emerged on a planet that was highly volcanic, with a huge amount of hydrothermal activity. The organic molecules were imported through carbonaceous meteorites, all produced in situ on the Earth through processes in the atmosphere or in the crust. The atmosphere was anoxic, it was mostly carbon dioxide. There's a high flux of radiation, but these were apparently just the right conditions for life to emerge. Now let's have a look at other rocky planets in the solar system. Mars and Venus, for instance. We know there was water on Mars. Venus, there is so much debate as to whether Venus started off wet or dry, whether it had water or never had water. We don't know yet. Anyway, so Mars didn't have oceans and protocontinents, but it had large areas, impact craters filled with lakes. We don't know about Venus. It had volcanic activity, hydrothermal activity, organic molecules. We're still finding them with, for instance, some instruments on the Curiosity rover in Gale Crater is still finding organic molecules of extra-Martian origin, and possibly also molecules that are produced in situ. It had and has still a carbon dioxide atmosphere and a high degree of UV radiation. Were they just the right conditions for life to emerge? We think so, otherwise we wouldn't be having missions to search for life on Mars. What about the icy satellites? Satellites like Europa, Enceladus, and others. Yes, we think that they are potentially habitable because they have water, they have oceans, they don't have protocontinents, they don't have any continents. Volcanic activity, difficult to say, but certainly we believe that there's some form of hydrothermal activity at the base of the oceanic coating. Organic molecules will have been imported by meteorites, etc., to the surface of the ice covering the planets, and by some means we think transported into the oceans. And in any case, there would have been organic molecules produced not by atmospheric processes, but by crustal processes. We don't talk about an atmosphere associated with these planets, and high UV radiation or any radiation, but it doesn't affect the possibility of life emerging in the ice-covered oceans. We don't know, but we're still hopeful. So the conditions elsewhere on other rocky planets and icy satellites were not very, very good, perhaps with the exception of Venus, but we really don't know anything there. There appears to be no possibility for primitive life forms to have evolved as they did on Earth. So the conclusion is that emergent life forms on other bodies in the solar system would have remained primitive and perhaps similar, but not identical to early life on Earth. And this means something similar to our anoxygenic anaerobic chemotrophs and possibly anaerobic phototrophs. So this is where I turn back to early life on Earth. And we look at the rock record. We have this nice sketch of the evolution of life from the NASA Astrobiology Institute. But in this area this is where we are going to be studying, passing the next half hour or so, let's have a look at what is left. Well, the whole of the Hadean, just about the whole of the Hadean epoch has been completely recycled, destroyed. It's lost. There are some zircon crystals and some geochemical signatures, isotopic signatures that give us some indication of what might have been there, but it's basically lost. We don't have rocks from that age. Other rocks, younger, the Eoarchean rocks are very highly metamorphosed. There are some indications of life in these rocks, but they're very highly metamorphosed. And so it's only as of about 3.5 billion years ago that we start getting rocks that are preserved well enough to be able to study these ancient traces of life. So let's have a look more closely at them. What do these rocks tell us? Well, first of all, the environment. I'll talk about the environment a bit later, but keep in mind we're looking at an anoxygenic Earth. Basically, very little oxygen, a little bit of free oxygen produced by abiotic processes, but basically anoxygenic. But the traces of life that we find in these rocks are extremely well preserved, and they are quite diverse. We find phototrophs, organisms that obtain their energy from sunlight. We find chemotrophs. There are two types. Excuse me, for the biologists of you, please excuse my simplifications of all of this. But in a simplified way, we can say there are the chemolithotrophs. These are organisms that obtain their energy by oxidation of inorganic substrates, such as hydrogen, carbon monoxide, sulfur, nitrogen compounds, and divalent iron, for instance. There are also the chemoorganotrophs that obtain their energy from oxidation of organic compounds. Where are these rocks preserved? Well, there are some ancient rocks preserved in Isua, in Greenland, also in Canada, at the edge of Hudson Bay. These rocks are very highly metamorphosed, as I mentioned earlier. But the two main regions for studying these ancient traces of life are the Pilbara Greenstone Belt in the northwest of Australia, and the Barberton Greenstone Belt in eastern South Africa. This is what the areas look like today. The Pilbara, it's basically a desert. This is a photograph in the winter, midwinter, Australian midwinter, so there's a little bit of green. It's an area called the North Pole because the Australians have a very interesting sense of humor. It's the hottest, one of the hottest places in Australia, which is why it's called the North Pole. Anyway, the North Pole is quite flat with some hills. The Barberton Greenstone Belt, also similar age, lovely mountains, just on the edge of Eswatini, the kingdom of Eswatini. But both of these areas, although they're different today on different continents, they have their ancient rocks were formed in very similar environments. On the left, we see cross-bedded volcanic ashes deposited in shallow waters, coastal waters, at the edges of a volcano. And from the Pilbara also, same situation, cross-bedded volcanic ashes deposited in a coastal environment, in this place, an infilling mud channel. And we look at the traces of life in these two areas. Generally, just to put you into the picture, the environmental picture, I mentioned that the protocontinents were basically submerged plateaus with just the volcanoes peeking out. And this is a sketch to illustrate this kind of situation. The largest deposits of rock are lavas. Lavas of one kind or another, mostly basaltic, some felsic lavas. They are coated with volcanic sediments, illustrated here in this dotted brown layer, covered by water. But there's a huge amount of hydrothermal activity associated with volcanism, especially the eruptive periods of the volcanism. So volcanic hydrothermal vents, micro hydrothermal vents to large-scale hydrothermal vents coming up everywhere. And a continual import of extraterrestrial materials, UV radiation, rain of volcanic ashes, etc., etc. So, I want to zoom into the coastal areas of these volcanic regions. The coastal areas, I'm showing you these photographs of the mud flats in the Bay of Mont Saint-Michel because they're quite illustrative of the environments that I will be showing you from the early Earth and in which we find traces of life. So if you take a spade and cut into these ripple-marked mud flats here at the edge of on the coast, you see this cross-cutting structures. These are typical of the cross-bedding. And we see this in the rocks from both Barberton, where you can see the ripple marks here, fossilized ripple marks here, and also in the Pilbara, where we see the fossilized ripple marks. So this is the environment. And these rocks contain traces of life, similar to the microbial mats that are shown here, or the cellular fossils that are shown here. I want to take a few minutes to explain how these traces of life can be preserved, because if we're looking at microorganisms, you'll think, well, they should just disappear, but they don't. They leave traces. So let's have a look at this. The most common trace fossilized trace of past life concerns the organic molecules that made up the organisms. On the left here, we've got a sketch of the red blobs are supposed to be colonies of microorganisms, large colonies of microorganisms, or you can have thin films of microorganisms forming around volcanic particle surfaces. We've seen this, we'll show you this, both of these. But then upon death, they degrade, their organic molecules degrade, they're mixed in with the sediments, and the organic molecules start breaking down. They break down into chains, aliphatic chains, or in aromatic structures. These are polycyclic aliphatics. These are the aromatic structures. And they break down. And to a certain extent, we can go back in the Earth's history and we can analyze the breakdown products and relate them back to what they were originally, the molecular component that they were originally. But I think the oldest known bona fide biomolecules don't go back to more than 1.6 billion years. We're talking about rocks that are 3.3 to 3.5 billion years old. So, but we still have the breakdown products, and you'll see that we can use these. Another form of preservation is just by, if the organism is robust enough, it can just be squashed, and you can see sort of the squashed remnants in fine-grained sediments, such as these features that were found in the fig tree formation by Emmanuel Javot in 2000, published in 2010. But these are very large structures. So you can have a look at the scale bar here. We're talking about over 100 microns in size. And so in order to survive, as squashed remnants, they have to be large and quite robust. And then there's the case where the microorganism can actually be fossilized in the sense that a mineral precipitates around the edge of the organism and or impregnates it. And it's the cellular structure is preserved. How does this happen? Well, if you can imagine that this is the external membrane of a microorganism. It's full of functional groups that act. Imagine it's like a hedgehog, and these functional groups attract minerals that are in solution, for instance, silica. And once the minerals have started precipitating around the edge of the microorganism, this is a micro-thin section of a microorganism. The scale here is 0.1 micron, and the dark spots are where minerals are starting to precipitate around the edge of the organism. And this carries on in a chemical way by polymerization to produce a crust around the organism. And of course, the organism dies and the organic matter degrades. And we've had a look at this with various experiments, myself and former PhD students, François Orange and Frédéric Gaboyer. But not all organisms preserve in the same way. This is work from François Orange during his PhD. He looked at two thermophilic archaea, Thanacaldococcus yanashi. This is a scanning electron microscope image of the organism. This is what it looks like. It looks it's not a nice looking organism. It is naturally rough, has a rough outer cell wall. But it also produces a lot of polymer, extracellular polymeric substances. So these are two images before fossilization. After fossilization, after 1 week, well, this is one microorganism. It is obviously dead. It did not like the process whatsoever. However, the EPS was fully fossilized, very easily fossilized and preserved. Now, let's have a look at the other organism that we use, Pyrococcus absci. This is a nice looking dividing cell. It's got a nice smooth surface. It's not ugly. And during fossilization, after 4 months, we can see that the cells have been coated with silica. In this case, you can see this. The spotted deposit is silica deposit, but the blobs are the cells. The cells are nicely preserved. So two organisms living in a similar environment that fossilize or don't fossilize in different ways. I'm going to talk about the larger scale microbial remains, and I think you've had a number of talks previously about this, about these phototrophic remains. Phototrophs, again, are the organisms that obtain their energy from sunlight. So on the right, we have modern microbial mats, a sort of green-looking layer, pustular layer. This is a cross-section. This is from work done by former PhD student Kieran Hickman-Lewis. And there's been a lot of work done on beautifully preserved remains of these phototrophic microbial microorganisms, or microorganisms. For instance, Nora Nofka has previously spoken to you about MIS, microbially induced sedimentary structures, which form mats, roll-up structures that are very well preserved on the surfaces of sediment. Abigail Allwood did some lovely work on a carbonate platform, 3.4 billion year old carbonate platform in the Pilbara, with different kinds of stromatolites preserved related to different microenvironments. A lot can be learned from these structures. Again, Kieran Hickman-Lewis, looking at other stromatolites from the Pilbara, 3.48 billion years old, has done some very nice in situ work, using synchrotron CT scanning to study the structure of these stromatolites in situ. And he's also worked on tabular microbial mats, such as these tufted microbial mats. This is from the Middle Marker Formation, 3.72 billion years old in Barberton. Looking at using FTIR, synchrotron FTIR, to look at the different types of molecules that are preserved, aromatic molecules, aliphatic molecules, the ratios between CH3 and CH2 groups, or CH2, CH3 groups, that gives some information as to what types of components have been preserved, whether they're microbial polysaccharides, or lipids. Sorry, I've lost my. Don't know what's happened. I've lost my point. It doesn't matter. And the ratio between the archaeal biomass compared to the bacterial biomass. And still with these tabular phototrophic mats, some of them can be preserved in exquisite detail. For instance, this is some old work of my own. I am allowed to talk about my own work as well. From the Westall chert, 3.33 billion years old in Barberton. It's preserved. You can just see the kind of films, filamentous film here. But it's so well preserved that we can take sections through it, study it as a modern microbial mat, look at the composition, the isotopic composition, distribution of light essential elements such as carbon, sulfur, and nitrogen, look at the structure and the composition of the organic matter to give us some information as to exactly what its biogenicity, basically. So there is a lot of evidence for phototrophic microorganisms in the Paleoarchean. They were widespread. Again, we emphasize that they were anoxygenic, and they formed microbially induced sedimentary structures. They formed three-dimensional stromatolites. They formed tabular microbial mats, and they formed biofilms, because this very thin biofilm here was only about 10 microns thick. They all developed on sedimentary surfaces in shallow water environments. So now I'm going to change tack and talk about the chemotrophs, the two kinds of chemotrophs. And I'll be showing you examples from two environments. One environment is influenced by hydrothermal activity. These will be the chemoorganotrophs. And the other environment are these sediments, but actually deposited on the coast, volcanic sediments. And there we'll look at chemolithotrophs. So first of all, the chemoorganotrophs and the hydrothermal environments. We're probably looking at something possibly not exactly the black smokers or maybe not exactly even the white smokers, but a kind of diffuse, some kind of diffuse emanations coming up from the sediments and the crust. And here we have some images of modern chemoorganotrophes colonizing hydrothermal environments, forming films and with a lot of extracellular polymeric substances. So we're going to have a look at an example from the Barberton Greenstone Belt from the Westall chert. And if the image on the left represents an outcrop where this vertical white line actually represents a hydrothermal vent that's cutting through layers and layers of sediment. The sediments are in vertical section. The laminations are like that. Well, they would be if you could see them. And this hydrothermal vent is coming up to the surface. The sediment surface would have been here. And the layers of sediment have been so thoroughly influenced by hydrothermal silica that they are black. And what we see in these sediments, we can see the very fine layers, but we have to use microscopy to be able to see them. And these fine layers are comprised of clots, dark clots, and the clots are actually carbonaceous. They have very spiky structures. They're stellate structures. They're obviously formed in situ. This is work with Kieran Hickman Lewis. He showed that in the center of these clots, there's always a volcanic particle. And we think that the volcanic particles were first coated by chemolithotrophs, because they're the organisms that use inorganic sources of energy. And then once the chemolithotrophs had exhausted the source of energy, they would have been colonized by chemoorganotrophs that used the organic carbon, the organic remains of the lithotrophs. Let's have a look in more detail at this. This is work that Kieran published in Science Reports in 2020. This is one of these stellate colonies. And Kieran looked at a number of aspects of these colonies, but especially we were interested in the concentration of transition elements associated with the organic matter. First of all, he used the PIXI spectroscopy. PIXI is proton-induced X-ray emission spectroscopy, or particle-induced. With this, we can see that at the center of the colony, there's always a volcanic particle that we can see here. But still associated with the organic matter, there are these transition metals that are important for microbial metabolisms, and because the early ocean was so enriched in these transition metals, any EPS would have chelated the metals as well. So we use this as a biosignature. Now we'll have a look at the other environments, the chemolithotrophs associated with volcanic sediments. I'll be looking at these here at the edges of the volcanic edifice. On the right, I've put some images of modern chemolithotrophs, Metallosphaera sedula, that have been grown on Martian meteorites. Just to give you an image of what they look like. This is work by the group of Tetiana Milojevic and Denise Kerbel published this. So let's have a look at this, and we'll be looking at samples taken from this particular rock. In fact, it was this hand sample here. These volcanic ashes that were deposited in a shallow water sedimentary environment. So first of all, the thin sections of thin sections of this rock show that it's full of these volcanic particles. Volcanic particles are often quite dark around the edges, And Raman spectroscopy has shown that there's carbonaceous matter associated with the edges here in green. Now, looking at the same sample with the scanning neutron microscope, you can see the volcanic particle outlined here. We'll zoom into this particular area, and we can see that there's a kind of carpet of small bead-shaped structures here that is rich in carbonaceous matter. And in fact, semi-bulk carbon isotope signatures of this material give a carbon isotope C13, delta C13 value of about -27 per mil, which is consistent with life. If we look more in detail at this carpet, you can see that the edge of the carpet here, abutting the phyllosilicas that are the replaced remnants of the volcanic particle. But you can see two size ranges, a smaller one, a larger one. They are in cell division. There's a kind of film coating them all. And further investigation of the rock shows magnificent preservation of a dividing cell here, more dividing cells here of different sizes. Again, dividing cells here of different sizes. And one in which one part of the dividing cell had obviously lysed and then was silicified. All of this, we can see that it's slightly embedded in the ground mass. However, I started working on this material in the year 2000, and my work has been criticized on the basis that these structures, they're very simple. They could possibly be abartic silica spheres. You can see some examples of abartic silica spheres here, or some kind of organic balls with this mineral coating, and these are from experiments showing similar cell division, etc. So the question remains, and also are they really biological or not? And there's another factor to keep in mind. This was the hand sample that I used for preparing the samples that I used for scanning electron microscopy. Well, how did I prepare them? Because how did I get from this flat rock surface to showing these nicely preserved structures? And no, we can't use a hammer and a chisel, as one would do with nicely preserved trilobites or dinosaur bones. So we have to use a corrosion process. So this image on the left shows the rock with the fossilized microorganisms inside. If you subject it to acid etching, and I would use hydrofluoric acid because this is quartz, the fossilized microorganisms are dirty quartz. So it's the matrix, this highly silicified matrix, that is etched preferentially, whereas the fossilized structures remain, as we see here. Now, the devil's advocate would say, right, okay, let's say that these are biological structures, but how do you know that they are syngenetic with the rock, i.e. they formed at the same time as the rock, and they weren't some kind of modern infiltration? So we have the problem of biogenicity and syngenicity. And this is where the work of former PhD student Laura Clodere comes in. She made a very, very painstaking study, multi-technique study of samples of this rock to demonstrate biogenicity and syngenicity. Yes, sorry, this was published in Astrobiology last month in February, so it's freely available. So she used a huge range of techniques to provide information, firstly about the environment, because we wanted to understand what kind of environment these microorganisms lived in, and the various analyses, especially analyses of the rare earth elements, demonstrated that this was an anoxic environment influenced by marine waters as well as riverine outflow, and that there was hydrothermal influence. But looking at the edges of these volcanic particles, again, the green here represents carbonaceous matter. Well, Laura used synchrotron DVD, deep ultraviolet fluorescence spectroscopy to study exactly where and what type of organic matter was there, revealing the presence of aromatic peaks, aromatic molecules, as well as, of course, the mineral peaks. And she also used Fourier transform infrared, as did Kieron, to look at the distribution of aliphatic CH stretching compounds, as well as the ratio of the various aromatic and alkenic compounds that are associated again with the organic carbonaceous matter that's here in green. As with Kieron, looking at the C2 to C3, C3 to C2 ratios, we were able to go back, say that the degraded organic molecules may have been related to aliphatic biopolymers, such as the isoprenoid chains of archaea. And also the CH3 to CH2 ratio gives us some indication that there were more organisms of the archaeal lineage than the bacterial lineage. Looking at using transmission electron microscopy to look at the carbonaceous matter, she was able to determine that there were two types of carbonaceous matter, or amorphous carbonaceous matter, as well as partially graphitized organic carbonaceous matter that we can see. Oh dear, I've lost it again. That we can see here with the with the diffusion spectra. Now, the presence of both amorphous and graphitized carbonaceous matter was also determined by the Raman spectrometry, which showed the D peak, which is more amorphous, and the G peak, which is more graphitized. So we have a mixture of the two types. But this is also what to be expected in ancient material of this age. So the conclusion is that the organic matter associated with the volcanic particles in thin sections of the Kiddie structure is biogenetic and it's syngenetic, and that a huge amount of complementary analyses at all scales was necessary to demonstrate this. And this is what we'll be needing also to work with materials coming back from Mars, for instance. So, but there is one thing, all of this work was done in situ on thin sections from the Kiddie structure, but they're not exactly the same as the purported microfossils that we see on the cut surface of the chert that were not in thin section. Are they biogenetic? Are they syngenetic? Well, stay tuned. This work is ongoing. So just to underline the importance of study of the early terrestrial signatures for extraterrestrial life, we'll have a quick look at the situation for Mars. For instance, early Mars, pre-Noachian Mars, Noachian Mars, there was water at the surface, maybe not as much as is shown in this artist's impression. Today, it's a dry desert. There would have been prebiotic molecules on early Mars, perhaps protocells, maybe some organisms similar to the chemotrophs I've been showing you. And if in the most ideal situation, perhaps something organism that had developed some kind of photosynthesis. Today, what do we have? We still have these prebiotic molecules raining down on the surface of the planet. And in the crust, if there's still life on Mars, it will be likely to be in the crust. It would have to be something similar to terrestrial chemotrophs, and again, totally anaerobic. Same thing for the icy satellites. So this is why the anoxygenic chemotrophs and possibly phototrophs on Earth are good examples or good analogues for what we might find on other bodies in the solar system. This is my last slide, and this is the team. Laura Clauderet, Kieran Hickman-Lewis, Frederick Fouché, who's our superb research engineer, Barbara Cavanazzi, University of Bologna, Tatiana Milojevic, who's taken over my group, my husband, Daniele Marcazzini, who's been with me in the field and is a bit sick of geology. Well, not really. So I'll finish my talk on that. Thank you very much.